Good morning. My name is Pieter Roelfsema, and I'm going to share my screen. So I'd like to thank the organizers to organize this symposium on brain and mind. And I'm going to present some ideas on how neurons in different brain areas of the visual cortex, interact with each other and how they contribute to conscious perception. So I'm going to ask the question, how does a visual stimulus reach consciousness? What needs to happen? And I'm also going to address the so-called binding problem. How do neurons coding different image properties, how do their representations get bound into coherent object representations? Okay, so after my talk, you should have some insight into these two topics. This is the overview of my talk. So first, I'm going to give a very brief introduction on the distinction between what we call feed-forward and feedback processing in the brain. Then I'm going to talk about the neural correlates of visual awareness. And then I'm also going to address so-called perceptual organization. So this is really about the binding problem. How do image elements, how are they bound in coherent object representation? And in the last, say, 10 minutes of my presentation, I'm going to say a few words of how we're now applying these ideas in an attempt to create an interface with the brain for blind people, a prosthesis. So I'm now going to start with a very brief introduction. So we're studying many of these processes in the macaque brain, although I'm also going to present some data from mice. And this is a very famous slide from many years ago by Velleman and Van Essen, who gave all these areas involved in vision a different color. Okay, so all these colored areas are involved in vision. Information enters in primary visual cortex here at the end, the back end of the brain. And then there are connections from V1 that distribute the information to all these other areas, these higher areas. And Velleman and Van Essen arrange the areas into a hierarchical scheme that is visible here. So here V1 is at the bottom, so this is retina LGN, then V1, and then there are feed-forward connections that propagate the information from lower to higher brain areas. And it's known, of course, that early areas of the visual cortex extract relatively simple features, such as the orientation of a light ball, and that neurons at the intermediate levels of the hierarchy, say area V4, represent feature constellations of intermediate complexity, and neurons at relatively high areas represent object categories, and of course also many other things. Now what I will demonstrate today, that once this feed-forward processing phase is over, information processing is not over, because there are also these feedback connections that propagate information in the opposite direction, here in blue, and also lateral connections between brain areas at the same hierarchical level. Okay, and one of the topics I'll talk about today is that they contribute to a process that we call perceptual organization, where all the image elements that belong to the same perceptual object are labeled with enhanced activity, thereby solving the binding problem. Okay, so now I will start with the first part of my presentation, which is on visual awareness. Of course, a big question, and the question that we asked is, what does stimulus need to do to reach perceptual awareness? And I want to start with a very simple task. So you should now look at this red dot, fixation point, and what I'm going to do is present a stimulus around here, and the only task is to kind of register whether you see the stimulus. So if you see the stimulus, you might want to raise your hand. If you don't see it, don't raise your hand. Okay, so 1, 2, 3. Okay, I suppose that most people saw this stimulus. And now I'm going to make it more difficult. And this is a stimulus that might only be visible for the good viewers. So 1, 2, 3. Who saw the stimulus? I'm going to try it once more. 1, 2, 3. And the last time, 1, 2, 3. Okay. Now, I suspect that many of you only saw a stimulus on the first and on the third one. And that is correct because on the second and fourth presentation, I did not present the stimulus. But sometimes it happens that people see a stimulus that was not there. It's called a false alarm. Now, this task is quite well described by the so-called signal detection theory. And in the signal detection theory, there is a theoretical construct that is the internal signal representation, or the strength of the internal representation, which is thought to be a stochastic variable. So if there's a stimulus present on the screen, then across trials, there is a distribution of this internal signal strength that is here indicated by the green distribution. So if I present a stimulus, you on a particular trial, you get a certain internal strength of the representation. And then the observer has to decide, did I see a stimulus or not? I'm going to report yes or no. And in the signal detection theory, there's sort of a threshold. And if the internal representation of signal strength is above the threshold, the observer is going to say, yes, I saw it. It's called a hit. But it may happen that the internal representation is weaker than a threshold. It will be called a miss because the subject says no. Now, a similar thing happens on the trials without the stimulus. So then you get, on average, a weaker representation of internal signal strength. And typically, if it's below the threshold, the subject would say, no, I didn't see a stimulus. It's called the correct rejection. But occasionally, the subject might actually report seeing a stimulus that was not there, and that's called the false alarm. Okay? Now, suppose that there is a stronger stimulus. So that means that the green distribution will shift to the right. If you still apply the same threshold, you would still get quite a number of false alarms here. That's the area under the black curve to the right of the red dashed line. So on such trials, on such blocks of trials, subjects might want to raise the threshold so that you get fewer false alarms, and it will not go at the expense of a drastic increase in misses. Okay, so this is simply a better threshold setting if the stimulus is stronger. But if you use this threshold on trials with a weak stimulus, of course, you will have quite a number of misses. That's the area under the green curve to the left of the threshold. Okay, so this gives you some intuition about the signal detection theory. It's an old theory, but the question we asked is, where is then this internal representation of signal strength? Where in the brain is it? And second, what causes the variability? Basically, what is the width of these distributions? And it turns out that our data supported a theory that is known as the global workspace theory about consciousness. And the theory makes the following, proposes the following idea. If you present a stimulus, there are feed-forward connections from lower to higher brain areas that propagate the information to higher areas, say of the parietal and frontal cortex. And the idea is, if the signal that arrives there is strong enough, then here there's a process that is called ignition. And I'll say a few words about what ignition probably means in the brain later. And then it definitely requires recurrent interactions, so feed-forward and feedback connections, that can uphold a representation of the stimulus even if it's no longer there. It's basically responsible for bringing the activity into a persistent state so that it can be reported later even if the stimulus is no longer there. Now, if you present a weak stimulus, the idea is that on some trials, it's so weak, it does not, is not strongly enough propagated to the higher areas to give rise to ignition, and that would be a miss. And another trial is propagated strongly enough, and then it reaches the threshold for ignition so that it can be stored in a persistent manner and can be reported at a later point in time. Okay, so that is the theory that we set out to test, and we basically trained monkeys on the same task you've just been performing. Okay, so the monkeys looked at the fixation point, and on 50% of the trials, we presented a brief stimulus, took it away, and on those trials, the monkey was supposed to make an eye movement to the previous location of the stimulus. Okay, it would be a hit. On the other 50% of trials, we did not present a stimulus, and on those trials, the animal was supposed to make an eye movement to this, what we call reject dots, thereby reporting that he did not see the stimulus. And if he made an eye movement to the reject dot, that would be a correct rejection. Now, across trials, we varied the contrast of the stimulus. So we started with a strong contrast, and the animals were basically on the staircase, and we gradually reduced contrast until they didn't see it anymore. Okay, and this shows you just the accuracy of one of the monkeys as function of the contrast, and we got quite nice psychometrical functions as expected. And the purpose of this slide is that we kind of sorted the data because we defined stimuli that gave rise to an accuracy better than 80% as easy. And easy we defined as this above this threshold. I already mentioned that 80%. There was also a low threshold, and if it was between the two thresholds, we said this is a stimulus that is of intermediate difficulty. And if it was below the low threshold of 40%, then we said this would be a difficult stimulus. Okay, and then we recorded from different hierarchical levels of the visual system, the primary visual cortex, area V4, which is a higher cortical area, and the dorsolateral prefrontal cortex, which is an area of frontal cortex that is actually involved in the generation of eye movements. So I should make a disclaimer, we are not going to dissociate conscious perception from the plan to generate a saccade here. Okay, and when we started to do the recordings, we were in for a surprise. I didn't expect this result. The result was, what we did here, we're recording now from area V1. The animals have electrodes chronically implanted in their brain, through which we record a multi-unit activity. And we see here the activity listed on difficult trials, intermediate trials, and easy trials. So as expected, you get better responses for stronger stimuli. But we also sorted the trials here based on whether the animals saw the stimulus or not. And actually we made sure that in this comparison, there's not a difference in stimulus strength. So these two responses on difficult trials, they were matched for stimulus contrast. And what we saw is that the stimuli that would eventually reach consciousness elicited stronger responses, that's the green response, than the stimuli that did not. Okay, as if on these missed trials, there is already a loss of information on the route from the retina to the primary visual cortex. You see that there is also quite a big difference in the strength of the response elicited by intermediate stimuli, but you see that the fractional difference actually becomes smaller for the easy trials. Okay, and what is interesting too is that the strength of the response on missed easy trials, that's this red peak here, is actually at least as strong, even stronger than the response elicited by the seen stimuli on difficult trials. Okay, this implies that a certain level of activity in V1 is no guarantee that the stimulus will reach conscious awareness. Okay, so the implication is that the information can also get lost downstream from primary visual cortex. We also recorded from area V4, and here the story is very similar. Okay, so again, a big difference between the seen trials and the missed trials. So again, information got lost en route to area V4. And also here we see that the easy missed trials actually elicit almost more activity than the difficult seen trials. So also a certain activity level in area V4 is no guarantee for a stimulus to reach conscious awareness. The situation was different when we recorded from dorsolateral prefrontal cortex. And now again, I repeat that this is an area that is also involved in the generation of eye movements. And what you see here is that there's sort of a categorical difference between seen trials in which the monkey is going to make an eye movement toward the receptive field and the missed trials in which he will not. You shall also see that the missed trials with stronger stimuli give rise to a little bit of activity in prefrontal cortex, but apparently it was not enough to reach conscious awareness. And here you can actually put a threshold, as if this activity is above the threshold, the monkey will make the eye movement and report seeing the stimulus, and if it's below the threshold, he will not. Okay. It's also interesting to look at the activity that was elicited on the trials without a stimulus. Okay, and you see in V1 and V4 there are subtle differences. So actually the false alarm trials in which the monkey reports a stimulus that was not there give rise to more activity than the missed trial, than the correct rejection trials in black. But in dorsolateral prefrontal cortex, you see there's this ramping of activity on false alarm trials where the activity at some point reaches the same level almost as on the seen trials. Now we cannot prove it, but we'll show it in the model later. This is well explained by spontaneous ignitions. So what might be the case here is that at some point activity in prefrontal cortex is relatively low, and at some point it reaches a high level, and this high level may be reached at different time points in different trials. Okay, so if you then average many of these trials with such a ramp, but these ramps happen at different time points, then you get this slow ramping activity if you average across trials. Now, why we believe that this might be the case is that on these trials without a stimulus, there's not a natural time zero. Time zero is actually a time where we would have presented the stimulus had we presented one, but we did not. Okay. And of course, then if the activity reaches this level, then it's quite compatible with what is predicted by the signal detection theory in prefrontal cortex. Okay, so if a certain threshold is reached on the hit trials or on the false alarm trials, it might persist then as persistent activity, sort of a working memory trace. If this threshold is not reached on the correct rejections in black or on the mistrials, then the activity subsides and the monkey reports not seeing the stimulus. Okay, so the basic idea is that whether the stimulus will reach conscious awareness is basically reaching a threshold of activity in the frontal cortex. Now, I presented these data to Stan de Hane a few years ago, and he said, this is really cool, because this is precisely what I have predicted in my global neuronal workspace theory. He said, I can model this, actually already made models, but he then made a model and that is embarrassingly simple because it contained only 5 neurons. Okay, and his model had the feed-forward connections from the LGN to V1 to V4 parietal cortex and frontal cortex, feedback connections in the opposite direction, and there were also self-connections between the neurons within the areas. But in total, there were only 5 neurons, and they were stochastic. So if you then present a stimulus to the network, it will be propagated to higher areas, And what Stan did is put extra strong recurrent connections, so feedback connections and self-connections, in parietal and frontal cortex. And if then the activity in parietal and frontal cortex is strong enough, it will actually give rise to self-sustained activity, reverberating activity. Okay, so if you then take away the stimulus, the activity will stay. It's basically a working memory trace. And that would then be a scene trial. On other trials with the same stimulus, it might be propagated, but due to the stochasticity, it might not reach the critical level that will give rise to this reverberant activity. Okay, if you then take away the stimulus from the screen, activity will decrease, and that would be a mistrial. Now, if you then do the simulations and you look at the activity levels in primary visual cortex, you basically see that if you look at the hit trials, there's more activity than the miss trials, and that's due to the stochasticity of the propagation from the LGN to V1. If that propagation is already weak, then there's a decreased probability that this ignition will occur downstream. You also, of course, see that for stronger stimuli, you get stronger responses. You get the same effect in area before. And in the frontal cortex of the model, because it's part of these two areas that together determine the reverberant activity, the persistent activity, if it reaches a certain minimal threshold, then activity will become persistent, right? That's what you see here. And on the strong trials that are eventually missed, there is certain level of activity that reaches the frontal cortex, but it's simply not enough to trigger ignition. And you also reproduce the effects of stronger activity on false alarm trials. On these false alarm trials, there's always some stochastic activity in the network, and on exceptional trials, say 2 or 3% of the trials, this is strong enough to trigger ignition. Now, this ignition point happens at different time points in different trials. So if you then average across a lot of trials, you get this slow ramp. Okay, so this is the output of the model. And I invite you to now compare it to the data that we obtained. So you can see that we can reproduce very many of these data features with a model in which the brain consists of only 5 neurons. Okay, now this is a very simple task. And for this, you apparently only need 5 neurons. But I'll show you later that there are more complicated tasks, of course, as well, where you need more than 5 neurons. Okay, so let me make some interim conclusions. So what we propose now is that the variations in internal signal strength of the signal detection theory are actually explained by variations in the efficiency of feed-forward processing, the strength of the activity level that reaches the frontal cortex. We suggest that false alarms can be explained by spontaneous ignitions. And it is also evident that the global neural workspace theory gives an excellent account for how a weak stimulus may or may not enter into consciousness. Okay, so now we're going to switch gears and talk about perceptual organization. If you realize how a complex image such as this one is represented in the visual cortex, then it's intriguing if you ask yourself the question, how do we see that this, say, the nose of the zebra belongs to the same animal as this leg? What does not help in this solving the so-called binding problem is the fact that neurons in early visual cortical areas have small receptive fields. So perception actually starts with fragmentation. Initially, the image seems to be analyzed in a piecemeal manner. And then the question is, what are the mechanisms that put together all these small image elements into coherent object representations? Okay, it's a so-called binding problem. And what I'm going to try to convince you of in the next couple of minutes is that the visual cortex actually implements what we call an incremental grouping operation, where image elements that belong to a single perceptual object are labeled by enhanced activity, and thereby signaling that they belong to a single coherent perceptual object. Okay, so if this is the image that comes in, and if you're interested in the central zebra, then the visual brain creates a labeled representation where all those image elements are labeled with enhanced neural activity. And this is essential for your conscious perception of a coherent object. This is what we call the incremental grouping theory. Okay, so now feed-forward processing is not enough, is the idea. You also need feedback connections for this labeling operation, basically binding image elements into coherent object representations. And try to illustrate this now. So what I'm going to show you very briefly is a visual stimulus, and the challenge is to see what is there. I suspect that there are many people who have not been able to recognize the object because it presents a segmentation problem. Okay, if you look at it for a while, you probably see that there's a small bird, but it's very well camouflaged. Okay, so the visual system first has to basically group together all these image elements of the bird into a coherent object representation before you can detect it. Okay, so how does this work in the visual brain? Well, the proposal is that it requires these recurrent connections. And that this is the case actually was already evident from the work of Victor Lamme, with whom I collaborated a lot on this project. And already 25 years ago, he demonstrated that if you present image elements in the receptive field of a neuron in primary visual cortex that belong to a figure, as is the case here, that you get more activity than if you present the same image elements in the receptive field. If you look carefully, you see it's precisely the same image elements in the receptive field, but here they belong to a background. So you now first get a feed-forward response, but it does not yet discriminate between figure and backgrounds. You have to wait a bit, and then you see that image elements that belong to a figure start to elicit extra activity. Okay, so in recent years, we've been trying to find out what the mechanisms are for this enhanced activity. So how does this work in the brain? I would like to discuss the role of visual attention in this process. Then I would like to address whether this delayed response modulation matters for perception. I want to demonstrate that it depends on feedback from higher areas. And I also would like to say a few words about the role of inhibitory interneurons. First, I would like to address how this works in the brain. And it's always useful, if you're interested in a task like this, to look back in the psychological literature, because you will find many clues about how these things could work. And in this texture segregation process, the proposal in the literature has been that there are two processes. The first is a boundary detection process that is sensitive to the feature differences that occur at the boundary between figure and background, basically the change in orientation. And then there's a second process that has been proposed. It's called a region filling or region growing process. If you select image elements with a certain orientation here, say the left orientation, then you might want to co-select adjacent image elements with the same orientation because they're likely to belong to the same perceptual object. Okay, now what are the neuronal mechanisms that support boundary detection? It turns out that you want to put inhibitory connections between neurons tuned to the same orientation. It's called iso-orientation inhibition. Okay, so how does this work? Well, suppose you're a neuron in the background, you're tuned to the right orientation. Then you get a lot of inhibition from your neighbors also tuned to the right orientation because they're also strongly driven by feed-forward connections. Okay, so your response will be suppressed. If your receptive field is in the center of the figure and you're tuned to the left orientation, you'll also get a lot of inhibition from your neighbors. And only if you are close to the boundary with your receptive field, the inhibition comes from only one side, okay? Giving rise to a relative release of this inhibition. Okay, now what about region growing? Turns out that for region growing, it's nice to have iso-orientation excitation. Suppose you're a neuron, you're tuned to the left orientation and you want to enhance a response. Maybe because you think that image elements in your receptive field are relevant to behavior. Then you would also like to excite your neighbors tuned to the same orientation, because then the features they're tuned to are probably also relevant, until the entire figure gets labeled with enhanced activity. Okay. This is of course a bit of a conundrum, because for boundary detection you would like to have iso-orientation inhibition. For region growing you would like to have iso-orientation excitation. If you build a neural network, these effects will cancel. Okay, so the proposal is that iso-orientation is actually implemented in the feed-forward propagation, in the feed-forward scheme, and the region growing happens a bit later through feedback connections. Okay, now you can make this work in a model. I'll briefly allude to the main ingredients of such a model. We published that. So here's the image, a figure with the left orientation and background of the right orientation. Here are neurons tuned to the left orientation responding to the figure, and neurons responding to the right orientation that respond to the background. Of course, in reality, these two sets of neurons would be intermingled in a single retinotopic map. And in the feed-forward pathway, we implement the iso-orientation inhibition scheme. So you see you get extra activity at the boundaries. If you now sum the two maps, you see indeed that there's a selective enhancement of neural activity at the boundaries due to the iso-orientation inhibition. Now the trick is to not only do this in V1, but to repeat the same connection schemes at higher areas. And at some point you'll reach a level where the size of the receptive fields matches the size of the figure. So here is one or a few neurons that are well driven by the stimulus, the left stimulus, and they don't get any inhibitory input from their neighbors. Okay, so they will be very strongly active. Psychologists call it pop-outs. For a larger stimulus, this will happen at a higher hierarchical level. Okay, and now the idea is to use feedback connections to fill in, also at lower levels, the center of the figure. And for this, you can actually build in a particular feedback scheme that makes this possible. What is nice about this model too is that the prediction is that this boundary detection and pop-out always happen because it's just determined by the feed-forward connections. So it's what we call the pre-attentive process, which is known to be the case. And a prediction of the model is that this region filling process depends on attention, because it depends on the loop to the higher visual areas and then back to primary visual cortex. That is something we can test. So what we did is we trained monkeys on a task in which they either had to detect a figure. So this gray rectangle was not visible to the monkeys, just a receptive field, but the figure is shown here. So on figure detection days, the only thing they needed to do is make an eye movement to the center of the figure. But on other days, they could completely ignore the figure, because now they were supposed to make an eye movement to a larger red circle that had been connected by a curve. Okay. So the prediction is that if the animal is attending, then this region filling should be not so strong. Okay. And so on figure detection days, once more, the animal was supposed to make an eye movement to the center of the figure. On curve tracing days, he could completely ignore the figure. And the figures were stationary, but across trials, the figure position differed. So on some of the trials, the receptive field would be in the center of the figure, but there were also trials where it was on the edge or even on the background, whereas the features inside the receptive field were always identical. Okay, so this shows you averages across many positions of the figure. And if the receptive field fell on the background, you get a red response. And if it fell on the edge or on the center, we got these extra spikes. Okay, now I'm interested in these extra spikes. So what I'm going to do is to subtract the red response from the green and the blue response. And then you get this somewhat complicated 3D plot, but I'll step you through it. What you see here on the x-axis is the position of the figure relative to the receptive field that was varied across trials. Okay, so here at 4, the receptive field is outside the figure. At 2, the receptive field was on the edge of the figure. The figure had the size of 4 degrees. At 0, the receptive field was in the center of the figure. -2, it was on the other edge, and then -4, it was on the background again. Okay? And on this axis are these excess spikes. So basically the difference between the red curve and the other two. So what you see is at an early point in time here, you get an extra activity elicited by the edge. And you get the same effect for the other edge. And then a later filling in of activity elicited by the center of the figure. Okay? So early boundary enhancement and a later filling in of the center of the figure as predicted by the model. Now this was on figure detection days when the animal is interested in the figure. Maybe you might want to picture what is going to be the prediction of what happens on those days in which the animals are not interested in figures. The prediction is that you get still the boundary enhancement, but the region filling does not occur. And that is basically what we found. Okay, you see that the boundary detection is still preserved, but the filling in of the center of the figure is much less complete. Now we also recorded an area V4, which is a higher visual area. Here we're again looking at the excess spikes, and you see there is more what we call figure ground modulation on the figure detection days. You see also that in the center of the figure, the response enhancement is there early onwards. And of course, that is true because the receptive fields in V4 are much larger. They're basically overseeing much more of the stimulus. Early enough to help V1 fill in the center of the figure at a later point in time. Okay, let me summarize this for you. So basically what we see is that you present the figure ground stimulus, you get visually driven activity. And then early on, you get extra activity at the boundaries. And then even a little bit later, you get also extra activity at the center of the figure, but this enhancement in the center of the figure is strongest if you attend to the figure and much less so if you attend elsewhere, as if these non-attended figures are left in a somewhat more primordial state. Okay. Now, a big question that we always had in the lab is whether this figure ground modulation is important for perception, or is it just an epiphenomenon? I already showed you that we saw this in monkeys. We're now looking at another species in humans. We sometimes have the possibility to record from humans with epilepsy. And then we can record multi-unit activity. And a few years ago, we were able to record spikes from area V3 in a human. And basically what you see is also extra activity elicited by figures compared to backgrounds. And this happened here at an early point in time because here receptive fields were relatively large. And the good news is that we can also study it in mice. Okay, also their figures elicit more activity than backgrounds. And to demonstrate that mice can perceive figure ground stimuli, we are training them. We train them in a figure ground task in which you say a figure on the background. And if they see a figure on the left, they lick to the left. And if they see a figure on the right, they lick to the right. And they can do that. I mean, they're not stellar, but they're clearly above chance level. So here is just a contrast detection task. You see that the mice reach about 90%. If they need to segregate a figure from the background based on orientation, their accuracy drops to about 70%. And that's the same for a face-defined figure and also for a texture-defined figure. So they can do this. Okay, we were really excited because now we can use the very many powerful tools that are there to study the visual cortex in mice. Okay, so the first thing we did is record multi-unit activity from primary visual cortex. You also have to realize that receptor fields in mice are quite large, so typically about 20 degrees in primary visual cortex. This shows you activity. Now, here on the left in green, this is simply a response elicited by a high contrast stimulus, and the black one is when the high contrast stimulus appears outside the receptive fields, so that's not so surprising. What is nice is that in this situation, the receptive field either falls on the figure in red or falls on the background, but now here the image elements in the receptive field are always the same. So also in mice we see these extra spikes elicited by figures. Same for face-defined stimuli and for texture-defined stimuli. You can then look at the latency of these activity differences. So contrast-defined stimulus elicits activity after 60 milliseconds, basically the latency of the visual response. But the extra spikes elicited by an orientation-defined stimulus happens after about 95 milliseconds, and for a face-defined stimulus a little bit later, which is also true for the texture-defined stimulus. Okay, so we basically find the same neural correlates as we saw in monkeys. And that is really exciting, because now we can do a dream experiment, an experiment that we always wanted to do in the lab. And that is to switch off V1 activity. And to make that possible, we inject an optogenetic protein called GTACR2 that inhibits, knocks out the V1 response. So we can basically knock out the entire V1 response and see what happens to accuracy. But, and now it gets really exciting, we can also let through just the feed-forward response and knock out the late activity. And let through more and more of the late activity by postponing the optogenetic stimulus to a later point in time. Okay? So that's what we did. And if the mouse is in the task in which it only needs to detect contrast, we then, what you see here, is we switch on the optogenetic light very early on. So say at about 30 milliseconds. That is when the feed-forward response has yet to happen. And if we don't switch on the light, you see the animals have an accuracy here of about, 80%. If you knock out the entire visual response, they drop to a level of about 60–65%, not to chance level. As if V1 is not completely necessary to do this task. Maybe there are also routes through the superior colliculus. Okay, but the accuracy drops. And if you then postpone the light to about 62 milliseconds, you see that the animals are always almost getting back to normal accuracy. And after about 70 milliseconds, they're back to normal accuracy. So what you see here in the green line is the average, and the black and gray lines are the accuracy of individual mice. Okay, so apparently in this task, the mouse only needs about 70 milliseconds of the V1 response. Okay, actually less of the V1 response because it's triggered after about 50 milliseconds. So about 20 milliseconds of V1 activity is enough for ceiling performance. As if then the higher areas take over and V1 only acts as a relay station. Now in the figure ground task, we play the same trick. And you see that if we shine the optogenetic light early on, that the animals really go down to chance level performance, 50%. Okay, demonstrating that V1 is absolutely essential for figure ground tasks. And you also see that we need to postpone the optogenetic light to, maybe about 110, 120 milliseconds before the mice reach ceiling performance. So this is exciting because we now demonstrate that this late V1 activity is essential for perception. And in the phase-defined stimulus, we need to postpone the stimulus by an even longer time, maybe even 240 or 160 milliseconds. Okay, and so now you need really a lot of late activity in V1 before the mice can do it. So yes, these late extra spikes are necessary for perception. Now the next question we asked is are these extra spikes that we see in V1 during figure ground tests, do they depend on feedback? And to test that, we simply injected the optogenetic silencer in higher areas. Okay, basically, we are now able to produce a ring of inhibition around the primary visual cortex. What we then see, this is when we don't shine the light, so you get a feed-forward response, then the figure ground modulation. If you do shine the light, we silence the higher areas, you see that the feed-forward response is not affected. Okay, and that is true across quite a number of recordings. But if you look at the late response, the figure ground modulation, you see that there's an overall decrease of the effect. That's highly significant. Okay, so these extra spikes, this figure ground modulation in V1 depends indeed on feedback from higher areas. I'll say a few words about the role of interneurons. As you may know, there are different types of inhibitory interneurons in the brain, the so-called PV cells, parvalbumin positive cells, that are thought to be responsible for gain control of the cortical column. Then there are these so-called somatostatin cells, and Adesnik and Scanziani a few years ago suggested that they are quite involved in surround suppression, basically maybe causing the reduction of activity for homogeneous image regions. Okay, so they might be involved in this weaker response elicited by backgrounds. And then you have a very special species of interneuron, the so-called VIP neuron, that inhibits the somatostatin cell. And it has been proposed to basically inhibit this surround suppression. Okay, so it might be a disinhibitory influence that takes away some of the somatostatin inhibition at the location of the figure. So that's what we tested. Now that we have these mice that can do figure ground, you have strains in which you can selectively express calcium sensors in specific interneuron types. Okay, and the first type we injected the calcium sensor is a so-called somatostatin cell, and the maybe counterintuitive prediction is that the somatostatin cell, which is maybe suppressing the background, should be more active for the background than for the figure. So it should actually have reversed figure ground modulation, and that's what we found. Okay. So here the activity elicited by the background is stronger than that elicited by the figure. Now then we scrutinize the VIP neuron, which is thought to provide inhibition to the somatostatin cell at the figure location. So that one should have the normal figure ground modulation, which is what we observed. And we looked at the PV cell, and were surprised to see this, but actually it even had a very strong figure ground modulation. Okay, so more activity elicited by the figure than the background. And this is known from PV cells. They typically seem to do the same thing as the excitatory pyramidal cells. Okay, so pyramidal cells have more activity elicited by the figure. And right, this is the level of figure ground modulation on the x-axis. VIP cells do the same thing as the pyramidal cells. They are responsible for suppressing the somatostatin cells, which are indeed have weaker activity at the figure location. PV cells in this experiment even have stronger figure ground modulation than the pyramidal cells. Okay, so that's quite an exciting idea that at figure locations there are certain sets of neurons that go up in activity, but the somatostatin cell has this very special property that it's more strongly active by those image regions that are not so relevant to behavior. Okay, So I think we are providing insight into those processes that allow the visual cortex to label all those image regions that are relevant to behavior with enhanced activity. And I would like to propose that this mechanism can solve the so-called binding problem. Now it's also interesting to compare the results in the first part of my presentation, where I suggested if there is just a simple contrast detection task, is V1 acts as a relay station. It sends the information to higher areas, and it's the higher areas that determine whether it's going to be ignition or not, whether a stimulus reaches conscious perception or not. But this is not the case for tasks in which you first have to do figure grounds. So if you only present the stimulus quickly, you see there are stuff, but you don't perceive the bird. For conscious perception of the bird, you actually also need these recurrent interactions to reach back all the way to primary visual cortex. Okay, now in the last 5 to 10 minutes, I would like to say a few words about an exciting project that we recently started that paves the way towards creating an interface for blind people. In the world, there are about 40 million blind people. And here we want to build an interface with the primary visual cortex. And we know that if you stimulate neurons electrically, so you place electrodes in the visual cortex, and you stimulate cells there, then subjects, can also be a blind patient, will perceive a dot of light. It's known as a phosphene. If you have many electrodes, you can of course create many of these dots of lights. And you can then in principle also play patterns of activity, trying to elicit meaningful percepts, even in a blind individual. Okay, so this is the system that we would like to build. It's a camera that sends camera footage to a processor that translates camera footage into brain stimulation patterns, as I illustrated here. They need to be sent wirelessly to an interface in the brain that then sends current to electrodes implanted in the brain. Now we're not yet wireless, but we started to do a project with patterns electrical microstimulation. So basically what we did, we implanted 16 of these so-called Utah probes in the visual cortex of a monkey. And so this is, as I said, it's not yet wireless. So the monkey has a connector on the skull. These are the electrodes. And this is how we place the electrodes. So we tiled quite a large fraction of the primary visual cortex with these Utah arrays. Okay, and then you get the receptive fields of those neurons. And you see that we cover basically a relatively large fraction of the lower right quadrant of the visual field. And also in a second monkey. Okay, now these monkeys are not blind, so we can simply map their receptive fields. And that's what is shown here. So the monkey is looking at a screen. Here you see these arrays, of course, they're under the skin. And you see that if you present a light bar, you get a wave of activity across primary visual cortex. Okay, you can also map the activity pattern in the visual field. So now basically plotting the activity at the center of the receptive field of the neurons, and you see that the bar is associated with a trailing wave of visual activity. And of course, this delay is caused by the delay between the retina and the cell in the primary visual cortex. Okay, so that is when we're in recording mode. Now we're going to go into stimulation mode. So we train the monkeys to make an eye movement to a gray dot. Then at some point, we don't present the gray dot anymore. We just present stimulation at one of the electrodes. And this is what you see here. So all these green circles are the center of the receptive fields of the electrodes we can stimulate. And the one that will flash in white is the one that we're actually stimulating. And the purple location is the eye position of the monkey. Okay, so you see that this works reasonably well. Monkey is making an eye movement to a phosphene. Now this has been demonstrated before, but of course not for hundreds of electrodes. Of course, the big question is, can the monkey also interpret pattern brain stimulation? Okay, to find that out, we trained the animals to recognize 16 letters of the alphabet. And in the visual version of the task, they look at the fixation point, we present a letter, and then they get the choice menu, and they're supposed to make an eye movement to the letter they just saw. So here the monkey would be reporting the letter D. And then we stimulate many electrodes at the same time at those locations where the receptive fields are that will be activated by the letter. So here we're trying to directly imprint on the brain the letter A, hoping that the animal will indeed report the letter A. And the exciting news is that this seems to work. Okay, again, here are all the receptive fields. The ones that we're stimulating are shown in white. We try to impose the letter L, monkey reports letter L. So we're really excited that this now works. Okay, so it's all, of course, proof of principle. There's still a lot of work before we have a working visual cortical prosthesis. And with that, I would like to end. So in short summary, I think I've provided some insight here in how stimuli reach awareness, that this is different for simple stimuli and for complex stimuli. I alluded to the mechanisms that are responsible for perceptual organization, claiming that the binding problem is solved by labeling a set of neurons that belong to a single perceptual object with enhanced neural activity. And I've also alluded to how we're now using our basic understanding of the visual cortex to create a visual prosthesis for the blind.